Hello guys, magandang topic natin ngayon. So, samahin niyo ako. So, we are going to tackle basic methods in the molecular laboratory. So, magsimula tayo sa specimen collection, how it is prepared, and how DNA and RNA are isolated from your cells. So, first off is sample collection. Okay, so I hope na try niyo na magpa-swab. Okay. So based on the standards on COVID-19 testing, so upper respiratory specimen ang kailangan, okay? And all in all, sabi ng CDC, ang recommended upper respiratory tract. But by the end of the day, yan, the type of collection that you're going to do is based on the suggestion of the manufacturer of the test. Yeah. So all in all, you're going to follow the procedure how the manufacturer meant the test to be, diba? So sila yung creator, sila yung may alam kung paano yung tamang specimen collection, sila magre-recommend, okay? But for the sake of our study, we're going to use the CDC guidelines. So sila yung reference ko, okay? So usually, the specimen that are used were nasopharyngeal swab and oropharyngeal swab. So I read all about this, and actually the specimen with the highest viral load is nasopharyngeal wash or aspirate. Yan. But since this is easier to do, faster to do, yan, usually what they do is nasopharyngeal swab and oropharyngeal swab. So, pag-usapan natin, okay, marami pa kayong matututunan as we go on. So, how to collect and handle specimens safely. So, work within 6 feet of patient as much as possible. Syempre, may physical distancing pa. So, if there is the need for swabbing, yan, so you will work closer, okay? Pero as much as possible, during the queue or the waiting, 6 feet apart kayo. And maintain proper infection control. So there are a lot of ways for infection control. Example, one part is the administrative controls and the engineering controls, if you remember in our laboratory safety. So there should be physical barriers, correct ventilation in the laboratory, right way on the flow of people in the laboratory. So who gets to go in the triage? Yan. So who gets to go in the laboratory? If you're symptomatic, you should be in the triage. So triage usually is set apart, isn't it? Or it's a dedicated space outside the hospital. Hindi ba? So there are administrative control, engineering controls that are involved. Biosafety cabinets, the use of acrylic barriers, follow standard precautions. So the typical hand washing, treating every specimen as if they are infectious, proper good microbiological practice, if the need to sterilize things, or example, waste disposal, proper waste management, Okay, so medyo mahaba kasi, but hopefully, alam nyo na. And the use of proper PPE. So what they recommend is N95 or higher level of respirator, okay? So N95 talaga ang standard. But CDC is indicating that it's okay to use face mask in limited resources, okay? But if the administrative, the management of the hospital is generous, dapat N95 talaga. Yan. So guys, I think it's good to remind you about fit testing, okay? So annually, this is performed in the hospital. Everyone in the hospital who are using PPEs, they should be annually fit tested. Dapat annual to, okay? So because not all brands would have your specific size, your fit, ganyan. So when we say the key factors required for a respirator to be effective, sabi niya the respirator must be put on correctly and worn during the exposure. The respirator must fit snugly. So it's snug. There's no gaps between the user's skin and respirator. The respirator filter must capture more than 95% of the particles from the air. Yan. So how to hold it. Yan. So donning. Yan, to make sure it's fit. Yan. Tapos, yung nose clip, yan, make sure that it's fitting. So, dapat, annually talaga, you should have, OSHA requires the initial respirator fit test to identify the right model, style, and size respirator for each worker. Ito yung napaka-personalized ng PPE, kaya nga personal protective equipment. So, how to collect respiratory specimens? So, dito lang tayo magpo-focus sa. So, the common specimen is your nasopharyngeal swab and your oropharyngeal swab. Guys, simulan natin. Notice lang itong nasopharyngeal swab. The Q-tip or the swab goes straight. Ayan. Straight po. So, we're not performing nasal swab kasi ang nasal swab, pataas. Notice ang nasopharyngeal swab, diretso. Okay. So, yun yung gusto kong i-emphasize. So, itong nasopharyngeal swab, NP and OP, oropharyngeal swab, ang doon tayo mag-focus. And I love this JF. So, first is your timing. Yan. So, ang maganda sa PCR, guys, that day 1 up to day 7, that you have your fever, up to day 1 pa lang, is you could have your PCR at pwede nang ma-detect, okay? Because there is already virus shedding. Yan. So, the virus is already multiplying. Yan. There are already remnants of RNA in the swab or in the specimen. However, yun, antibody detection, to produce those antibodies against those genes of the SARS-CoV-2, kailangan after 17 days, after 2 weeks pa. Yan. Okay rin naman ang antigen detection, no? Nagiging accurate na rin ang antigen detection. Kasi even the first 4 days, yan, pwedeng maging positive sa antigen detection. In fairness, our test kits are improving. So, collect as soon as possible after symptoms begin. Ooh, okay. So, PCR, based on this image, so, during the incubation phase, yan, hindi ka pa magpa-positive sa PCR on the first day of the symptoms. So, mahirap po yung asymptomatic. Okay, so collect as soon as possible after symptoms begin, within 7 days, and before antiviral medication, and collect multiple specimens on multiple. That's the best. Yan. Parang kunyari specimen, stool collection. If you want to test for parasitic infection, mas maganda multiple collection, hindi lang once. Yan. So for our upper respiratory tract, let's focus on nasopharyngeal swab first. Okay. So use synthetic swabs or thin plastic or wire shaft. So usually the kits already include those types of wire shaft. Actually, first time ko nakakita ng wire shaft na ito ngayong SARS-CoV-2, okay, that have been designed for sampling nasopharyngeal. Yan. Do not use calcium alginate swabs or swabs with wooden shaft. Yan. So yung typical na cotton buds natin, it's not good, okay, because it's calcium alginate. Yan. Or read the container of the swab, okay? Because some of this, gayong ganito yung itsura, it could look like perhaps it's rayon or dacron. Pag rayon, dacron, okay lang. So calcium alginate, it's a no-no. It may inactivate some viruses. And swabs with wooden shafts, okay? So if both the nasopharyngeal and oropharyngeal swabs are collected, combine them in a single tube, okay? To maximize test sensitivity and limit the use of testing resources. I'll show you later a journal na mas maganda daw pag pinagko-combine yung NP and OP specimens. Okay, so calcium alginate swabs are a no-no. Yan. So, look at the OP swab, yan. It looks like a calcium alginate. However, mukha siyang rayon. Yan. Or, itong NP swab, yan. Mukha rin siyang flux swab. Yan. So, do not use calcium alginate or cotton swabs or wooden sticks. They may interact with your PCR test. Yeah. So specimen collection, guys, is important, di ba? Maintaining quality specimens is part of producing quality results. So here, how do you perform nasopharyngeal swab? Tilt the patient's head back 70 degrees. Insert the flexible shaft mini swab through nares parallel to the palate. Yan. So guys, diretso. Yan, diretso, diretso. Walang angle, okay? So, diretso ang kamay. Yan. So, it's not going on an pa-up na angle, okay? So, diretso ang, so, parallel daw o parallel. Okay, it's not in an angle, okay? Pag pa-angle, nasal swab yun. So, ang ginagawa natin nasopharyngeal. So, dapat straight. Okay, if it's an angle, straight. And we completely insert, okay, until there is a resistance, okay? So about 2 inches. Distance is equivalent to half of the distance from the patient ear to the nostril. So if this is the full distance of between the nostril to the ear, yan, half. Yan, mga 2 inches. So gently rub and roll the swab. Leave swab in place for several seconds to absorb secretions. Pero usually parang may experience ang bilis nila ginagawa. Slowly remove the swab while rotating it and immediately place in sterile tube. So usually yung viral transport medium, VTM, containing transport medium. Yan. So in a seated position, tilt the head back at 70% angle. Yan. So I think that kulang pa yung tilt, pero pwede na rin. Yeah. So straight yung swab. Okay? So until you will meet a resistance. So parang ang layo ata nang napuntahan mo siya doon. Ano naman yan. Usually ganyan lang. Sa gitna, mga 2 inches. Sa half daw eh. Halfway. So, ilalagay sa VTM. Yeah. So, oropharyngeal swab. So, we swab on the tonsils and the posterior oropharynx. Yan. Ito yung tonsils. And ito yung tonsils. Ito yung posterior oropharynx. Yan. Yan. So, then, place it on the transport tube. Tip first. Okay, the tip ng swab. So here, the comparison of NP and OP swabs for the diagnosis of 8 respiratory viruses by real-time reverse transcription assay. So yung conclusion dito, na it's best. The most effective approach is the combined NP and OP specimen. Class notice, ang maraming authors. Grabe yun. Pwede pala yun. Ganun karami. So storing and shipping respiratory specimens. So if we cannot process it immediately, up to 3 days, yan, store it at 2 to 8 degrees Celsius. However, longer shipment, talagang negative, 70 degrees and below, okay? And guys, if it's already an extracted nucleic acid, yan, nawala na yung mga contaminants like proteins at negative 70 degrees or lower, okay? The typical. So, hindi kailangan long hour, matagal, okay? So, kung na-extract na siya, kailangan freezing talaga. So, guys, ito, hindi to red top tube, okay? It looks like a red top tube. But you have to read the label, okay? It's a viral transport medium, okay? So, this is where your swabs, yan, would be suspended. Yan. So, dyan pinasok yung swab tip. Yan. So, dyan mo i-reconstitute yung upper respiratory specimen mo. So your BTM is good for transferring virus, chlamydia, so bacterial yan, mycoplasma. So those which needs a cell culture, yan, chlamydia and mycoplasma, mahirap yan i-grow sa mga culture media. Kailangan cell culture. So when we have a cell culture, so the microorganism will grow inside of the cell, 'di ba? Kasi nga example virus, 'di ba? It needs a host cell, okay? Hindi siya nako-culture sa isang nutrient, agar. For diagnostic tests and molecular biology techniques, yan. So what are the components of VTM? Actually, your VTM is standardized, okay? According to the specified standards of WHO and CDC. So ano ba dapat daw ang laman? Which should have calcium, magnesium, heat inactivated fetal bovine serum, FBS, and antibiotics like gentamicin and amphotericin B, so there would be no microbial growth. Okay? So guys, notice itong FBS, fetal, so as in fetus, bovine as in cow serum. So how do they collect this serum? By getting fetal blood from pregnant cows. Yan. So itong FBS, so it's a growth supplement for the cell culture. Yan. So it's talagang they harvest the blood of cow fetuses. Tapos, yon, they get the serum. Alam niyo naman siguro how serum is acquired. Hindi ba, ipapaklat mo yung blood tapos you will centrifuge it tapos maghihiwalay yung cell component saka yung serum. So, yon guys, pregnant cows. So, it's good that you're going to read about it. And pero some farmers, they wouldn't know na yung mga they do it ethically, they try it. And pero syempre hindi naman natin masabi kung they really follow the rules, 'di ba? Pero yan, they try to do it ethically. So they get it from fetal cows. So bago i-slaughter ang isang pregnant cow, yan, kunin mo na nila yung blood ng fetus. Yan, guys. Yan, so, yun. Isa sa magandang natutunan ko dyan. So, unacceptable specimens. So, if improperly identified, the request form and the container does not match the identification or no identification at all, insufficient volume, I don't know. Yan, siguro may spillage, yan. Kaya naging insufficient yung volume. Especially kung upper respiratory kasi hindi naman siya volume talaga, di ba? Improper specimen type sent. So, siguro baka pag sputum ganito, ganon, or saliva. Spillage, specimen shipped at improper temperature. So, dapat -70 kung matagal. Excessive specimen transport time. So, ito na yung bulk ng topic natin. How do we prepare our specimen? So, by isolating your DNA and RNA. So, extracting the DNA and RNA. So, for the SARS-CoV-2, what we extract is the RNA virus. Yan. But we'll start with DNA, okay? So, DNA isolation or extraction is very important. It's most critical because what happens here will downstream. Ayan. So, downstream procedures and analysis relies on the quality of the DNA. Okay? So, in our quality control, every step of the processes should be correct, okay? From the specimen collection now to the isolation, okay? If you did not isolate well the DNA, then you will not be amplifying anything in the process of PCR, okay? So, kung nasira yung DNA during your process or wala kang na-acquire na DNA, diba? So, what's the point on proceeding to your PCR? So, mahalaga ang DNA isolation, okay? So, what happens in DNA isolation is you separate your DNA from the other cellular materials such as protein and membranes. So basically, anything that is not DNA, kailangan matanggal. Just like your proteins, your cellular membranes, lipids, polysaccharides, any other biochemical compound na hindi DNA, kailangan it would be precipitated off. Kailangan matanggal. Okay? So yun ang pag-aaralan natin. How would we acquire only the DNA? Isn't it that the DNA is a very small compound, hindi ba small siya? So makikita natin ano bang itsura ng DNA macroscopically. So what else? So the rationale is we remove inhibitors that could interact with our PCR, and we want to get a stable solution of high quality DNA, okay, that can be stored for prolonged duration without degrading. And so, you know, guys, DNA is important for murder investigation, di ba? For checking yung mga ancient na DNA. Yan. So, if you want to store the DNA of Cleopatra, yan, or kung sino important person, di ba? You want it, we don't want it to be degrading over time. So, yun yung mga pag-aaralan natin today. So, there are so many sources of DNA. So, DNA plasmid, so from bacteria, viral nucleic acid, so DNA viruses like Hep B, and DNA from blood and biologic fluids. So usually pag blood and biologic fluids, yung WBC yung source natin ng DNA because it has a nucleus. Tissues and cells, forensic samples, plant and fungi. So example, itong plant and fungi, they have cell walls. And also bacteria, so kailangan i-crush or i-homogenize so that you could free up the DNA. Yeah. From food and feeds and ancient DNA. Ayan, archaeologic specimens. So generally, ito nangyayari. Okay? You have to lyse your cells so as you could free the nucleus and the DNA. You have to precipitate the DNA and also precipitate the protein. Okay? So usually yung protein, it stays at the bottom and the DNA floats at the top of the tube. Yan. So kailangan mong hiwalayin. Now you could resuspend or rehydrate your DNA yan, for storage. So since hiniwalay mo siya, so it's not floating anymore on an aqueous solution, or kailangan mo siyang i-wash, i-purify, tapos i-rehydrate mo. Yan. Yan. So 3 basic steps. So first one is lysis. Yeah. So lyse, what do we break down? So lysis is the breaking down of cells. Okay. So we break down the cells so we could release the nucleus and we could release the DNA. Okay. So how do we do it? First is mechanical disruption. So pwedeng manual method. Yun nga, using a mortar or pestle, a homogenizer. Yan. So, kumbaga parang blender inside the laboratory. Or you could use detergents and enzyme. Like, organic pala to. Sodium hydroxide. Yan. And SDS, sodium dodecyl sulfate, if I'm not mistaken. Yan, tris EDTA or TE, EDTA, and SDS. Yan. Okay. Hindi pala organic. I mean organic extraction. So usually in the organic extraction, yan, ito, base, sodium hydroxide, SDS. In the inorganic extraction, ito yung ginagamit for cell lysis. So here. Yan, precipitation. So you separate the DNA from the cellular debris or the protein. Okay. So what are dead? So sodium ions to neutralize the negative charges on the DNA, which make them more stable and less water soluble. So normally, your DNA has a negative charge. So you want to neutralize it, okay? And guys, alam nyo ba, yun nga, take note your DNA has a negative charge. So the scientists, yung mga scientists, ang galing nila. Because isn't it that opposite charges attract, okay? So, ginamit nila yung negative charge ng DNA. So, they would get a bead a bead with a positive charge. Tapos they could magnetize, kumbaga, yung DNA. So that's a way of extraction sa other methods. So take note that DNA has a negative charge. So that it would be neutralized, pwedeng lagyan ng sodium ions, okay? And make it less water soluble para mag-precipitate, okay? Para hindi mag-dissolve. And we could also add ethanol. So ethanol is a dehydrant, diba? Or an N-isopropanol. So alcohol to, these are dehydrants. So it could cause also the DNA to precipitate out of the aqueous solution. Yun. So para mahiwalay yung DNA. Yan. Para mag-precipitate. So ethanol and isopropanol ginagamit. And purify. So, pag nag-precipitate na yung DNA, the DNA could now be extracted to purify it further para mawala talaga yung mga contaminants. Linisan mo with an alcohol or a buffer. Yan. And you could redissolve it or resuspend it in water. And later, I'll show you images of that. So, let's get to know first the reagents used for extraction. So, we have cell lysis buffer. So usually, the manufacturers already has prepared it. So I'm just going to introduce. Ano ba yung mga contents? Just give you an idea. Detergents, guys. Yan, buffers, EDTA. Di ba? Ang galing. Ang use ng EDTA. So, designed to lyse the outer cell membrane but will not break down the nuclear membrane. The nucleus is still intact. Ang galing ng lysis buffer na, guys. So, ito yung mga extraction yan, para mawala yung mga proteins like phenol and chloroform. So, it will allow the proteins to settle at the bottom, itong phenol. And chloroform prevents the cutting of DNA during isolation. Hindi maputol. It solubilizes lipids and a lot of protein to remove them from the DNA. So partner actually ang phenol at chloroform. So when you use it, usually the contaminating proteins, the lipids will settle at the bottom and the DNA will float at the top of the solution. Okay, so bottom yung magsisettle sa bottom yung mga ayaw mo, tapos yung protein mag-float sa taas. Yan. So it's easier to extract or isolate the DNA. Yan. So proteinase K, yan. So in other procedure, kung hindi phenol or chloroform ang gagamitin mo, you could use proteinase K. Yan, remove most of the protein. Yan, to remove the protein by digesting with proteolytic enzymes. So enzyme method ang proteinase K. Active against broad spectrum of native protein. So it's good for removing proteins. Tong proteinase K. Powerful siya. Yan. And guys, EDTA, ang raming use ng EDTA dito sa DNA analysis natin. So it inactivates the DNases. Ito kasi yung mga DNases to, it destroys or damage the DNA. So you have to remove it. Yan. So EDTA as is in the removal of DNases. How? By chelating magnesium ion. Yan. So magnesium ion is important partner or cofactor of DNase. So kung na-chelate mo na tong magnesium ion, so the DNase will not work anymore because it requires a cofactor. Okay? So not only calcium, yan, but also magnesium ang kinekelate ng EDTA. So it envelopes your magnesium ion, so it makes your DNases inactivated, okay? Because it's an important cofactor partner. Ito, sodium dodecyl sulfate, SDS. Yan. It's also a detergent. So it's a lysis reagent. Okay. Helps the cell membrane and nuclear envelope to break open. What's good is it also inactivates your nucleases para hindi masira yung DNA mo or RNA mo. So, di ba, these are enzymes that could destroy. So, ito, trace EDTA, buffer. So, this buffer, guys, is important in resuspension for storage of DNA. Kung na-extract mo na, it's good for storage. Yan, because it prevents DNA and RNA degradation. It's used for final resuspension. Yan. It preserves the DNA and the RNA because it's a buffer. So it prevents the extreme changes in pH. So 95% ethanol and isopropanol, so they are also used in precipitation. So to ethanol and isopropanol, for inorganic precipitation, yung phenol at chloroform, organic precipitation. Ay, ganun. Ethanol, organic. Isopropanol, inorganic. So, phenol rin. Okay. So, una nating aralin, guys, ito, partner phenol and chloroform method. So, chemical base muna tayo. Tapos next is the salting out method. So we use salt. And the proteinase K method or the enzymatic method. And we have silica column based DNA extraction method or the solid phase. So what they use here, guys, are silica column or silica beads. Yan. And apparently, yan, itong silica column, silica beads, most probably it has a positive charge. Yan. So silica particles has affinity for DNA. Kumakapit itong DNA sa silica. Ayan. Namamagnetize. Parang gano'n. It has an affinity. So it's a way na to isolate your DNA. Kumakapit siya. It loves, it sticks to the silica column. Galing. So the advantage of organic extraction, kasi ito yung una nating topic, rapid denaturation of nucleases. So itong nucleases, it destroys your DNA and RNA. So at least it will be denatured, matatanggal, and it stabilizes the RNA. So if the RNA is, so guys, in the isolation of RNA, actually, phenol and chloroform, pwede pa rin gamitin. So it stabilizes your RNA. Drawbacks or disadvantage, yun, manual intensive and difficult method. Yan. So the one that's commonly used in the laboratory is actually silica column based. Yan. Sa RNA isolation ng COVID testing. Yan. Okay, so simulan natin. So basic steps, lyse the cells, open it up. Yan. So that you would free the nucleus, yan, your DNA. And we extract by the addition of phenol and chloroform, then centrifuge. Okay, so once you centrifuge it, the proteins, the lipids will settle at the bottom, and the DNA will rise to the top. Okay, top aqueous solution with precipitated DNA, and bottom protein contaminants and cellular debris. And so precipitated DNA will be purified or rehydrated or resuspended with water or buffer. So ito yun, no, precipitated. Yun yung itsura, guys. So hindi ba pag ganito na siya, mas madali nyo nang i-separate. Yan. So what we added in the lysate, so nung after na nilagyan mo ng buffer or detergent or enzyme, yan, you add phenol and chloroform, okay, and centrifuge. Maghiwalay ang lipids, proteins, at DNA, looking like this. So now you could separate it, place it in another container, add water, or clean it further, yan, by washing it with buffer or with alcohol, tapos resuspended it with water. Yan. So, vortex with phenol, yan, plus chloroform. So, your chloroform is good for stabilizing. Tama ba? Balikan nyo lang yung centrifuge. So, it will look like this. So, the upper layer will have the DNA. Sa gitna pala is the proteins. The organic layer, phenol. Kasi nga, di ba, ang lipids, ganun, cell membrane, gawa sa lipid, it will dissolve sa phenol, diba? Kasi organic compound siya. So, sa middle pala, yung proteins. Okay. So, ganun rin, guys. So, you have your cells here, right here. Kunyari, your VTM. Yan. So, you added lysis buffers or lysis reagents. We have sodium hydroxide or SDS. Yan. So after the release of DNA from the cell, further purification requires the removal of contaminating proteins, lipids, carbohydrates, cell debris through high salt, low pH, and phenol and chloroform. So, nag-add ka ng phenol and chloroform as your extracting reagents, di ba? But you could increase the salt level and the low pH. Yan. It increases the yield and the purity of your DNA. Yan. So, kumbaga, di ba, may mga add-ons. Yan. So you could acidify it, yan, through the addition of a bit of acetic acid and the salt. Yan. So now there is the cell debris, yan, at the bottom. Okay. So once you added extraction reagents, yan, so DNA will in the top part, the proteins will be at the bottom part. Yun. But at the end, you could also add, kung DNA lang ina-isolate mo, you could add an RNAs to remove RNA contamination para DNA lang puro. Yun. And then resuspend. So how about the salting out method, ma'am? So kung sa phenylchloroform ang lysis reagent, sodium hydroxide, ito inorganic DNA na to 3 EDTA, and SDS naman yung lysis reagents. Okay. So to free up your DNA. So what they use is isopropanol and salts. So different salts. Yan, to increase the DNA extraction. So pag inorganic, ang ginagamit isopropanol. Okay. So same thing, the proteins will settle at the bottom in this area or lower part of the solution, and the DNA will stay at the upper part of the solution. So now you could resuspend. Yeah. Inorganic DNA or salting out method. So you use salts and also isopropanol as your extraction. Pwedeng isopropanol lang. Yun. Next is proteinase K method. Yan. So actually proteinase K nga, maganda talaga siya pangtanggal ng mga proteins. However, yun, the proteinase K enzyme has low stability. So pwedeng masila siya agad. Yan. It's labile. Yan. High yield talaga. However, it's time consuming. Also, it should be in the cold chain. So, dapat, it's processed in a cold environment. Yan. So, yun. Yan. So, example, ito, may blood, tapos yung PE buffer, trace, EDTA buffer, yan. So, mix well. So, itong buffer na to ginawa niyang lysis buffer. To pellet, so usually, yung tawag doon, yung na-release, yung precipitate, ang tawag usually pellet. Add buffer again. Yan, para matanggal talaga. Malayes talaga siguro. Yan. Tapos add your proteinase K plus DNA extraction buffer. Ano kaya tong buffer na to? DNA extraction buffer. Perhaps the reagent that is provided by the manufacturer, isoamyl alcohol, and salts again. So this increases the yield. Wash with ethanol and dissolve DNA. So wash with ethanol. So further purification na to. Yan. Sabi niya, time consuming, however, high DNA yield. Marami kang makakollect ng DNA. So this is good. Especially in research, in experimentation, in forensic. Kung kunyari talagang 1 drop of blood lang yung nakuha mo or talagang tuyo na yung dugo. Yan. So, you need really to extract as much DNA as you could. Ang galing rin, no? Kunyari ng mga forensic na kasi splatter of blood. Yan, konting blood lang. Tapos, they're expecting that you would acquire the DNA. Diba? So, next is silica-based. So, the gist of this, guys, is DNA is attracted to silica. Kumakapit siya sa silica. And so, silica-based method works on the unique chemistry between silica and DNA. Silica is positively charged and it binds or adsorbs the DNA. So, the word is adsorb. with your negatively charged DNA. So, example, itong malaking beads na to, guys, is your silica. Itong black na representation na to, it's the DNA. Okay? So, your DNA, yan, holds on. It is absorbed by your silica. Ang galing, no? So, yan. First is you lyse your cells and spin. Yan, to remove the large debris. Yan. And usually, the debris will settle always at the bottom, hindi ba? Apply lysate reagent to silica column or B. So once you have lysed your cell, yan, so you could now apply it to your spin column. So kung narinig nyo na yung spin column, that points to your silica column or your beads. Yan. Saan na ba tayo? Yan. So dito na. So addition of alcohol and acidification to remove contamination. Okay? Kumbaga mga add-ons para to increase the purity, para mas magandang extraction, add ka ng alcohol, di ba? Because these are extraction reagents, and acidify it. And do a microcentrifuge or an automatic nucleic acid extractor machine. Yan. And so kumapit na dito yung DNA, yung black. Yan. So kumapit na sa spin column. Now you would get a clean tube and you will wash yan, the elute. Yan. So ano kung ano yung na-absorb sa silica column, guys? Since na-absorb, kumapit siya. So now you need to get a buffer to wash it para matanggal mo yung DNA sa silica column. Okay, wash it. The adsorbed DNA will be washed using buffer. And itong may drop dito, so those are the elute DNA. Yan, wash it with your buffer, and what will be left? Ito yung natanggal na kumapit sa silica column. This is now your purified DNA or your elute DNA. Yan. So yung elute, meaning it's the extraction of the adsorbed DNA. So yan yung adsorbed DNA, okay? Elute. So yun yung term. So widely accepted because of its good quality DNA yield and minimal simple operating system. Yan. So what you're just going to add is your lysis reagent, yung mga, kung may addition kang extract, yung acid or alcohol, but it's not, I don't think that's necessary. Basta may DNA ka, may silica column ka. So, nabibili itong lalagyan. Guys, binibili na nakaganyan. Okay? What you're just going to place is the lysate na lang. Yan. So, kung anong kumapit dito. So, my friend said that this extraction could last up to 30 minutes. Yan. So, matagal lang yung microcentrifugation because you want to make sure that the DNA is adsorbed by the silica column or beads. Okay. So, ang hirap maghanap ng image, guys, kasi nangyayari ito sa loob na lang eh. So, ang end product, nakikita mo lang is the elute. Okay. So, you wash it with a buffer. So, para matanggal mo yung DNA na kumapit sa silica at ma-isolate mo siya, kailangan mo siyang i-wash ng buffer. Okay. Yan. So, actually, ito yung ginagamit na ng kaibigan ko na may nucleic acid extraction or tawag nila, since COVID testing na, RNA extractor machine. So, other methods for extracting your DNA. So, we have DNA extraction using an ionic resins. Hindi ba pag anion positive charge? Hindi ako nagkakamali. Or, tama. So, yeah, let's check. I searched it a while ago. I forgot. Cations positive charge, anion negative charge. Okay, whatever. Okay, calyx positive charge naman. Binds to negative charge DNA. So, parang the same thing with the silica, iba lang. Calyx ang tawag. So, calyx, it's a patent. So, your calyx absorbs your DNA so that you could extract your DNA. So, it's a type of resin. So I don't know why anionic resins. And magnetic beads. So magnetic beads magnetizes the negatively charged DNA. Okay. So notice meron na namang word na elute DNA. So you will get the adsorbed DNA. Extract the adsorbed. Kung ano yung kumapit na DNA, you will extract it by washing it with buffer. Okay. So positively charged magnetic beads. So usually, kasi ang DNA, di ba, negatively charged. So kumuha ka ng isang positively charged. And they are separated under the magnetic field. Okay. So once the magnetic beads attracts the DNA, so tanggalin mo na yung mga proteins and they move it through a magnetic field. Okay. Okay, next, guys, we have CSCl or cesium chloride density gradient method. So just like a centrifuge, yan, you could separate the different materials in a solution based on their density. So example dito, sa Ficol density gradient centrifugation. So this is a procedure for separating particles in which the sample is placed in a preformed gradient such as sucrose or cesium chloride. Example, you have a blood sample. You add sucrose or cesium chloride, or the reagent is called Ficol. Upon centrifugation, yan, the macromolecules are bonded in the gradient. So they would go to their assigned positions based on their gradient, okay? So that you could acquire them. So ang source ng DNA sa DNA sa blood, guys, is WBC. So now you could get your WBC extract. So for DNA, itong CSCl gradient method, so your nuclear DNA stays here, your mitochondrial DNA stays here. Yan. So yan, based on density. For how do we store your extracted DNA in a very cold temperature? Yan, -70 up to -80. To prevent the nucleases activity. So that the DNAs will not degrade your DNA. So, ano pa? What else? EDTA is good for inactivating your nucleases. Yan. Diba the concept of magnesium? Because it chelates magnesium. The magnesium is the cofactor of your nucleases. Yan. And what else? So, yung temperature, you could add EDTA or you could store it using your trace buffer or your TE buffer. Now we are in RNA isolation. Yan. So you could pause and mag-break muna. So here, this part is how we would isolate your SARS-CoV-2 because it's an RNA virus. So take note, guys, na 80, most of the RNA that we could collect is your total RNA. And only 2.5 to 5% are your mRNA. Yan, ang konti niya. Pero mahalaga rin na extra kanyang mRNA. Mamaya, pag-usapan natin. So, guys, isolation of RNA is difficult. Yan, very crucial. So, because your RNAs is a major contaminant. So, kailangan may strict precautions to avoid sample degradation because RNA is volatile because it is easily attacked by your RNAs. Yan. So, our goal here is to prevent its degradation. Common siya ma-degrade. So, the common contaminant, your RNAs, yan. What's horrible about it? It's natural occurring. So, if you're not wearing the correct PPE, yan, if your hair goes to your sample, if a skin cell goes to your sample, it has RNAs. Yan. Bacterial and human source, RNAs could be difficult to inactivate because it can reactivate even after autoclaving. It must be eliminated or inactivated. Talaga, completely, and there should be an RNase-free area. So there should be constant autoclaving, cleaning, not reusing glasswares, always using gloves, PPE. RNF area, RNase-free area. So how do we protect the specimen from RNAs? Wear gloves at all times. That's not an exception. Hindi pwedeng magkamay. Just like, di ba? I mean, ako sa phlebotomy, yan, kung pwedeng hindi mag-gloves, hindi mag-gloves. Pero, yan, it's a good habit to practice always wear gloves, especially in isolating RNA. Use RNAs, free tubes, and pipette tips. So, as much as possible, reusable tubes and pipette tips are not recommended. Wag na. And yung RNAs, free tubes, and pipette tips, yan, could be bought, bilhin nyo. Use dedicated RNase-free chemicals. Yan. So, class, dapat, ayun, you have to read your chemicals. Kung anong nirecommend ng manufacturer, yun na. So, if ever you're going to use a glassware or a pipette, kailangan pre-treat materials with extended heat at 180 degrees for several hours and wash with DEPC treated water or sodium hydroxide or hydrogen peroxide. Supplement reactions with RNase inhibitors. And reusable glassware is seldom used. Matindi itong RNase na kalaban sa laboratory. Mas lang hindi mo pa nakikita. So this is an enzyme. Okay. That breaks down your RNA. So how do we extract total RNA? One common one is your phenol chloroform extract. Okay, so parang ganun lang rin. However, sa lysis buffer is performed in detergent or phenol in the presence of high salt and RNase inhibitors. We also add GITC, guanidine isothiocyanate, strong denaturant of RNases, instead of high salt buffer. Yan. So, maganda siyang i-add in the lysis buffers. Yan. RNase is precipitated by the addition of 2 volumes of ethanol and 1 volume of isopropanol. Or, yan. If you're going to use ethanol, 2 volumes. Or isopropanol, 1 volume lang, mas konti. So, always at the top, maprecipitate yung RNA, at the bottom, yung mga protein contaminants, yung mga lipid. So, RNA precipitate is then washed. Yan. So, sa final purification and resuspension, wash with 70% alcohol and resuspended in an RNase-free buffer or water. So, kung sa DNA isolation, we add RNAs para masila yung RNA. Kasi we just want DNA, correct? For RNA isolation, we add DNAs to destroy the DNA because we don't want DNA contamination. Yan. So, same thing with your silica column-based RNA extraction method or solid phase. Pareho lang yun na. So, ito yung, guys, sa loob nito, yung silica beads or column. Yan. So, dumikit na yung RNA sa silica beads or column, yan. Wash it, so with buffer. So itong lumabas dito sa may butas, guys, that's already your yung mga contaminants. And now you have an elute. So wash again with buffer. Yan, and RNA na. Okay. You use, yan, okay. So yan, wash it with buffer so that you could get the elute of the RNA. Yan. Okay na yun. So, guys, ito na. mRNA. So, your mRNA is only 2.5% to 5% of your total RNA. Okay? So, the principle so that you could get your mRNA, guys, what's unique about our mRNA? It has a poly A tail. Pag sinabi nating poly, marami. Pag sinabi natin A, adenine. So this is what your mRNA tail looks like. Yan, puro A. Yan. So what we're trying to do is how would we capture your mRNA? Because your A will be attracted to letter T. Yan. To an oligopolythymine. Or actually, it's also attracted to polyuracil, to a U. Pero usually, ang ginagamit natin, T. Okay? So, the bead or the column has 3' T, 5'. Okay? Kasi ang mRNA, poly-A tail. Yan. So, 5' muna to 3'. So, itong mRNA natin, attracted siya sa T. Okay? Kasi it's filled with A. Yan. So, ganun naman. So, kung sa DNA, negatively charged, attracted siya sa positive charge. Itong A, ang kahinaan niya, attracted siya sa T. Okay, poly-T. So, your adenine will form hydrogen bonds with your thymine. Yan. So, yan. After washing away the residual RNA, the poly-A RNA is eluted by washing the column. So, we could elute it, remove the adsorbed RNA using warm low salt buffer. Yun, sa DNA, buffer. Containing detergent to break the hydrogen bonds between the mRNA and the column. Okay, sabi niya, warmed low salt buffer with detergent. Okay, so ganyan ma-isolate yung mRNA. Galing, no? It's attracted to the poly-T. And why do we want to extract only mRNA? Why? Because your mRNA reflects or represents the genes that are expressed by the DNA. Okay? So, example, ito, may spike protein, yan. And the spike protein is, so, the mRNA with instruction for making the spike protein is developed in a lab. So, most of our vaccines, guys, are made up of spike protein or S gene. Yan. So, yung Omicron, guys, it lacks S gene. So, basically, our vaccines are not that effective against Omicron. Pero buti na lang Omicron, guys, it only infects the upper respiratory tract. It does not reach the lungs. So, kaya nagka-surge ng Omicron because it lacks S gene. Ayun. So, mRNA, so they created mRNA with instructions for making the spike protein. So, they isolated this mRNA out of your total RNA. So, using your poly-T bead or column. Yan. Para magkaroon ng mRNA vaccine. Yan. And so on. So, we have mRNA vaccines. And that's all. So, guys, that's our trip to isolation, specimen collection, preparation, and isolation. Thank you, guys, for listening. Bye, guys. Thank you.